have the pleasure to introduce Burton-Kelly, who will be presenting his PhD proposal project to us today. Matt has been with us for a little while. He's completed his master's here. He first came to us from St. Lawrence University in Canton, New York, where he completed his senior thesis on trace fossils, which has subsequently been published while he was here. He did a master's project on the morphometrics of analytical project on some unusual, well, not necessarily unusual, but some horribly simple clams at the KT boundary interval in southwestern North Dakota, which he presented both at national and international meetings, the international being in Antwerp, Belgium. He has presented on database management design at national meetings, and he's presented a little bit on this topic so far. He's going to present today. So, without more ado, Matt Burton-Kelly.
Thanks, Joe. Forgot to get the clicker out. Never mind the clicker. Before I get into any details about this project, I want to go over a few key points about how I'm addressing the problems that have come up. To keep you in the right frame of mind, here's what I'm interested in answering. How can we distinguish between morphologically convergent specimens? Most of the time, all we have to deal with is morphology. And by morphologically convergent, I mean similar in shape because the organisms live in similar environments. For this project, I'm looking at applied names or identification, not evolutionary relationships or phylogenetics. I'm interested in how fossil specimens are assessed once they come out of the ground, and then how these specimens are identified. What I'm not worrying about is anything higher up than the genus level. I'm looking at the genus names being used, and so the evolutionary relationships between organisms above that level don't matter for this particular project. So on this I have identification is not equal to classification. Classification deals with the concept of a taxon or a type of organism relative to other types of organisms and their relationships. This project focuses on applying names well and consistently to specimens. I'm particularly interested in making sure we apply the right names to things. It makes everything easier if names are applied consistently. At the very least, I think we need to formalize what identifications are problematic and whether that problem comes from ambiguous genus or species descriptions or from an incorrect identification. The issue you end up with is that there's no final arbiter, especially in the fossil record, of who's right and who's wrong, and so an incorrect identification in one person's eyes can be refuted using other criteria. Which brings us to the people problems. You can't get away from people because the specimens don't care what they're called. They're specimens, and the evolutionary relationships don't particularly care. And the problem is that we are people and we're applying names and relationships to things, and that we care and that we make mistakes. It seems like these problems are less problematic now than they were 100 years ago when people had less access to the wide variety of descriptive works, but there are still problems, people getting in arguments over things that they might not get in arguments. But even if we can't solve these problems, these people problems, at least acknowledging them will make it easier for people coming along in the future. So finally, we can get to the meat of the project, freshwater mussels. The focus is on the freshwater mussels of the family Hyriidae, which is now restricted to the southern hemisphere, specifically South America and Australia and New Zealand. And I'm focusing on this family because it's a medium-sized group compared to other related families, and there are some interesting names that are applied in the fossil record, which I'll explain. The problem with freshwater mussels in general is that they have convergent morphology. This is due to the plastic phenotype of freshwater mussels. So populations that are in similar environments develop similar morphologies. In addition to convergence among living taxa, like you can see here, living taxa from China and Alabama the populations develop these similar wing forms and so on across there. There can also be convergence among taxa that are separated by geologic time. To start thinking about freshwater mussels and geologic time, here is a taxon range diagram for all the genera in this order Unioneuta from the southern hemisphere. So there's 4 families represented here. The Hyriidae in the green names, Mycetopodidae in orange, the Iridinidae in red, and the Hyriidae in gray. So just focus on the length of those bars and not the thickness that measures a different quantity that I'm not talking about right now. And these bars are ordered according to first appearance date, going from left to right. So about half the genera have fossil representatives, and those are those genera that have bars. You can see the genera on the right, they just have a little blip at the top that means that they're extant only. And of those genera, a fair number of them are Hyriids. And of all the Hyriids, you can see that Diplodon on the left is one of those that has a very long range. This diagram shows just about the same thing, but for the Hyriids only. But here you can see the age of each fossil occurrence, and the little boxes mean the number of fossil occurrences within that age range, and then the thick lines are error ranges on those boxes, and the thin lines are just continuity between them. If you look at the distribution of occurrences of a genus through time, you can see some fairly large temporal gaps from last occurrence of, or you can see gaps between occurrences of fossil specimens to which names have been applied. And you can see that Diplodon over here on the left has this huge gap. And I'll highlight that here. And it has this temporal gap of specimens that have been called Diplodon of up to about 200 million years, depending on the error ranges, between the Triassic occurrences and the late Cretaceous and Cenozoic occurrences. There are 4 possible reasons for such a gap. The first is sampling bias or cryptobiology. The second is lack of concurrent deposits. The rocks just aren't there from that age. The third is taphonomy, change in morphology of the organism after it died, and so that's preventing us from identifying it as Diplodon. And 4, non-presence of the taxon in the first place. So the first 3 of those causes assume that Diplodon in this case was there all along and represents the null hypothesis that the lineage was continuous through that time. But the last hypothesis assumes that these 2 groups of occurrences can't both be Diplodon, and that the last cause represents the alternative hypothesis and means that it suggests that these are 2 different taxa, one early and one late. For my project, I'm interested in looking at all the temporal gaps in the Hyriid record, or at least for 3 genera, but I'm going to stick with the Diplodon example for right now. The null hypothesis here, as I just said, is that Triassic Diplodon is the same genus and the same genetic lineage as Cretaceous and Cenozoic Diplodon, and we're just missing part of the fossil record for a number of reasons. This maintains the status quo of the specimen identifications both before and after this gap. The alternative hypothesis is that the Triassic Diplodon is a different genus, a different lineage than the Cretaceous and Cenozoic Diplodon. There was one genus group during the Triassic, there's one genus group in the Cretaceous Cenozoic, but they aren't the same lineage. And they might even be closely related, but under this hypothesis, they aren't the same. And what I mean here by saying that the gaps in the fossil record are not real is that you can't have a gap in the fossil record for a single taxon if it's really two taxa. You have one and then you have the other, and they have no relationship to each other. To disprove this null hypothesis, I need to show that the lineage could not have survived in a suitable habitat during the time covered by the temporal gap. If the genetic material couldn't get from point A to point B, or time A to time B, the populations at A and B can't represent the same taxon or type of organism. So this is a very difficult thing to do, to disprove this by proving that the lineage could not survive, because it's impossible to prove a negative. Instead, what I'm hoping to do or trying to do is build a statistical model that evaluates the probability of the suitable habitat through time and space for the genera I'm studying. Methods. Into the matrix. The goal of this project is to build a model of the possible Hyriid habitat through time, then use this model to test the probability of the lineage of population A, which is before the gap, even if not preserved, surviving long enough to produce population B, which is after the gap. So to do this, first I need to take paleo environment into account, because we're dealing with long spans of time, and so the environment was not constant. For discrete time intervals of 10 million years, I'm determining the paleo environment for the regions surrounding known fossil occurrences. The paleo environments are based on previous landscape models, such as this one by Blakey, and direct literature and museum sources for taxa that are indicative of certain environments. So other freshwater taxa are indicative of freshwater environments during specific time periods. And this is a map of environments during the Miocene, or a model of environments during the Miocene. Second, I need to take paleogeography into account. For each of the 10 million year intervals, I will determine the relative position of the paleo environments and the fossil occurrences, taking plate tectonics into account. And so this can be accomplished through the point tracker software by SCOTES, which was used to produce this plate tectonic model of the Miocene as well. To simplify the data for analysis, I'm rasterizing it into 1 degree grid cells for each 10 million year time slice. This is occurring after I use the point tracker software to reconstruct the paleogeography, because I have to use point data to go into point tracker, which rotates it, and then I convert those points into rasters. And the Hyriid occurrence data are converted into a presence and absence raster. So there are two ways to compare the environments through time. There's a simple way and a hard way. The simple way is to compare the time slice data just by stacking the rasters, the paleo environment rasters, on top of each other. There are two questions to ask when comparing these time slices. So first, do the freshwater environments overlap? This would signify a continuity of habitat through the time interval for the same area. Second, do freshwater environments cross boundaries through time? In the case of headwater stream capture, you have one stream capturing the headwaters of another stream, which moves a section of freshwater habitat from one drainage basin to another drainage basin, and moves the biotic component as well. And I'll be looking for that on a regional scale. In the example here, I have, these are time 1, time 2, time 3, time 4. Freshwater environment is denoted by the green, volcanic or mountains are the orange, and marine is blue. And so as you go through time, you can see that these two freshwater drainages are separated first by a marine incursion, then by some marine and this volcanic arc, then by more volcanic activity between them, and then only in T4 do they join up, and you could have a transfer of freshwater organisms back and forth across this barrier. The more complex way is to do the same raster stacking, but using a niche modeling program to produce the rasters. So for example, this is a figure produced by GARP, genetic algorithm for rule set production. And what GARP does is takes a bunch of different rasters of different environmental data sets and compares the values on those rasters to the occurrence data for the taxon under study, and produces a model of the probability of the taxon being present in each cell of the raster. And it's essentially multiple regression for each grid cell. So instead of having a bunch of samples from sites that are unrelated, you have your sites arranged in a grid, and you're producing a probability model for each grid cell. And then those can be overlain. The rasters that are produced by GARP or another niche modeling program can be laid on top of one another, and you can do the same stacking as here and look at how the environments change through time, but you can take probability into account. On to expected results. Generally, freshwater mussel habitat will be available through time, but there will be certain geographical corridors that limit the dispersal of the taxon under study. Secondarily, these corridors will be studied to test their validity. And these are the points at which the dispersal of the population would be under greatest threat of extinction. I also expect to see certain boundaries that populations were probably not able to cross, at least at the scale I'm working. These are barriers that are interesting. If the barriers do exist to give support to the idea that more than one taxon was given the same name over time, and if the barriers are crossed by some groups but not others, then you can sort of think about why this filtering effect is going on. Possible conclusions. This project is expected to produce a useful method for questioning taxon identifications on opposite ends of temporal gaps in the fossil record. A comprehensive paleoenvironmental model with detailed geographic and temporal control is needed for better work to be done. This model, the way I've set it up, sometimes it feels super detailed in that I'm using 1 degree grid squares, and sometimes it feels like I'm drawing with a crayon, trying to mark where things were in the past. So more detail, more temporal control would be good. Unfortunately, models like this are subject to interpretation, my interpretation, and Joseph's and the rest of my committee's interpretation. But models are also good at being improved over time with more data. So the results, either accepting or rejecting the null hypothesis, give weight to either people trying to split taxa because that temporal gap is too big in their opinion, or trying to say that no, the taxon survived that entire time. Retaining a single taxon through time raises questions about longevity of freshwater mussel genera, and splitting a taxon is testable. If you find an occurrence, then you have to reevaluate the model and see where the problems lie. Thank you.